PCR plates, whatever, but 96 well plates. So I don't know what we've got 14. I think roughly 14 extractions we just did. So we could fill up 14 wells of this plate. And so the point is we can fit 96 of those samples on one plate, and then this is what we can use for all of our downstream next applications. So you can see that. Is that what you're kind of going for? And then beyond that, I was also going to say that we have a new piece of equipment in our not in our physical lab, but in the building that we are funding, where we actually take this whole process that you've just kind of been exposed to, and we actually have a robot that will do larger numbers of this in a much shorter time span, where we can do these 96 well plates in about 15, 20 minutes of what you've just seen. Technically, with all the setup work, it's probably about an hour, but nonetheless, we can extract a whole one of these and have it ready to go for everything we want to do next for our analysis work in half hour.
And just to give an illustration of how the technology has advanced over the years, when I was a graduate student 20 years ago, we did DNA extractions in this sort of a volume because the techniques to visualize those markers at the end required huge amounts of DNA. So imagine scaling up what you've got in that little tube into this thing, and the DNA looked like a big, the technical term is ball of snot. That's what it looked like. That's how much we needed. Now we're dealing with very, very small quantities of DNA using very sensitive equipment to tell if a particular gene is or is not in a particular plant.